till my presentation is loading.
Yeah, sure.
It's not loaded yet. Yeah.
Sure,
Yeah,
sir.
please,
Sure, sir.
please.
Once it's ready, sir, then we'll start.
Yes,
sure.
yes.
People are probably commuting, sir. That's why joining in few minutes here and there. 6 to 7, no, sir, the commute time.
Yeah, we can, one or two
Yes, sir.
minutes.
Yes, sir.
Dr. Lahari.
Yeah.
Permits. Yeah.
Yes, sir.
We'll start anytime you're ready. No problem.
Yeah, right.
Just a second. Ma'am, could you help me from where I shift to the
Sure,
thing?
sir. So on the top, you will see a share button. There's a leave button, and right next to it, there should be a share button.
Okay.
Yeah. Press on the share button, sir.
On the right, left.
On the top bar, on the top of your screen, there's a leave red color button. Right next to it, on the left hand side, there's a share
I
button.
can see one bar in the center, but nothing on the top. More actions.
No, next to more actions, sir, there will be a camera and mic button.
Yeah,
And
it's
next to
there.
that, share. Yeah, share. Press on share, sir.
Raise hand, show conversation, show
On the
participant.
right hand side, sir, camera and mic is there. Next to that, there should be one share button.
Open share tray. Okay,
Yeah.
got it.
So now press on screen or window or tab. Share
Yeah, got
content
it.
in terms
Got
of
it.
that. And then
Yeah.
now you can choose that you want to share the presentation. In window, it will come under the window.
Yeah, there's a window.
Yeah.
Okay.
Say share, and then it should say okay. So it will allow us to
Select a tab. Which one? Webinar. Okay. Share. Okay, my current screen.
Yes, sir. Your screen is coming in. 1 second.
Are you able to see my screen?
Yeah, we can
Okay,
see
that's
Google
great.
right now. Yes, yeah.
No, but now it must have come.
Yes, it's come now. So now you can.
Okay. Okay, something happened.
Right now we are seeing only your web browser, sir. Once your presentation is open, we'll be able to see that.
So has it come yet?
No, sir. Sir, did you only choose your web browser as the option?
Okay, just a second. Okay, there might be two options there. Okay, just a second.
Yeah.
Share.
उधर से तो मैंने किया. Sir, 1 second. What you do is press again share. Press again share.
Stop. It will stop sharing. Okay.
Yeah.
Then again
Now
I
you
should
start again. Go to share. Now
Okay.
press on screen or window or tab. Now press on window. In the window, you are getting your presentation option.
Window. Okay. Yeah. Here I have.
Yeah,
Okay.
yeah.
Microsoft.
So now press the
Okay.
presentation and say share.
Okay. Is it coming now?
Yeah,
Yeah.
we got
Can
it.
you just make it
It's-
full
Yes,
screen?
yes.
That's great. That's great. Okay.
Yeah.
Good. Yeah. Thank you. Let's come.
Okay.
So
Really
just
sorry
make
for
it
that.
full screen. That's it.
Yeah. I think now it must have come in full screen.
Not yet. Sir.
No, you need to click the.
Maybe
down.
some lag is there.
1 second.
Some cup is there.
Okay. One cup is there.
Bottom right side, you will see that.
Bottom.
There is hide and there is a stop sharing.
No, on your presentation,
Yeah.
bottom right.
Presentation.
Oh, this
Yeah,
cup.
that one. Yes.
Yeah, that one.
Okay.
Yes.
Got it.
Yes.
Is it full screen right now?
Not yet.
Not yet. 1 second. Not yet.
Maybe we'll just wait and it should come.
I think
Not
on your
yet?
screen, the slideshow is on the top, not at the bottom.
No, on my laptop it is. This is full screen.
It is bottom
slideshow
right.
icon is there.
Press again. I think. Try again.
Maybe the wrong window has been chosen. Just a minute.
No, we
I'll
are
go
seeing
back there.
your program.
Okay.
Just try again. Just press it again.
Okay,
maybe.
I'll just try once again. Maybe this time it should come. I'll make full screen and then I'll come back to it. This one is full screen. I hope now it's okay.
Yeah. It's
Yes,
fine.
yes. You got it.
Okay.
You got it.
Okay, so straight away we should start.
We can start. Yeah.
As you all know, this is a very important topic about microbiota and its relation to our health and disease. My topic today I shall cover under these things. Like firstly, I'll take you to the introduction, what the definitions of microbiota and what we define microbiota and various other bacteria which are found in our body or in any organism.
Dr. Sumit, 1 minute. We have introductory remarks by President, Chairperson. Okay. Yeah.
Okay, just a second.
Let me introduce the speaker.
Yeah, Dr. Lahari, I welcome you. Please, your introductory remarks. Yeah.
Good evening, everyone, and thanks for asking me to chair the session. It's my pleasure to introduce one of the very prolific speakers, Dr. Sumit Kumar Rawat from Sagar, and he is associate professor and nodal officer in Bundelkhand Medical College and Hospital, and he is at present working. It's near Bhopal, and his career objective is in infectious disease research, learning a long-term goal in making humanity to overcome through his research on various infectious disease. And he has been awarded. He has been working in different places, like he was a supervising clinical labs in virology, microbiology. He was a teaching graduate medical, paramedic postgraduates, and nursing undergraduates, and he has a long list of teaching associates, and his research interest is mainly diagnosis, prevalence, and immune pathology of infective hepatitis, HIV, infection re-emerging and emerging viral infections like SARS-CoV-2. And he has his past experience was working in different institutes, and he has been awarded plenty of awards. If I start reading all of them, probably it will be longer than his talk. So I would welcome Dr. Sumit to go ahead, and he's a well-known person with plenty of awards under his belt. So I welcome Dr. Sumit. It's all yours now.
Uh, Thank you so much for Dr. Lahiri sir for the kind introduction. And now I think we shall proceed with our continuing medical education. So I shall, my talk shall proceed with introduction, the various facts which are upcoming every year, every now and then with the microbiota. The microbiota and what is dysbiosis and its role in health and disease. Then various facts which are related to various skin diseases. Then how do we study and what tools we can use to study the microbiota. Then I shall come to the applications and the treatment approaches which have come up to use not only in various skin diseases but also various other diseases like maybe asthma, various systemic diseases, cancer and other digestive disorders. Then there are some limitations as of now, and finally I shall conclude. So this is very interesting that what we think of us as a human, like we usually think of only the human cells, but each human is a complex ecosystem and in whom the microbiomes also play various ecological roles. So if we talk of a pure human, probably we were purely human when we were in the womb, when we were not born. And as soon as we are born, we interact with the environment, and so many various commensal organisms and various environmental bacteria, they start interacting with us. And that starts from the birth canal, and later on the environmental air, and then the mother's skin and various food and whatever nutrition we are taking, it all determines the formation of our microbiome. So what we call a human is actually our own human cells. It is a lot of bacteria, fungi, archaea, viruses, and whatnot. New discoveries to be made. So the human is actually a human plus his microbiome. So, shall a doctor consider a patient a single human organism or a community of organisms? So, this is a new concept which is emerging. So, for a complete addressal to any problem, I think we should consider the whole community as a target, what we want to treat, and we should target to restore the balance. Whatever the balance is disturbed, only that is leading to various diseases. So, we should target to restore the balance whichever way we could. So till now we have been in a concept that most of the bugs are bad, and when you grow up you are always taught like germs are bad, and there are so many things like around you, various ads on the television like Kitanu everywhere, and they show in the Life Boy and Dettol advertisements all over that. So we have a concept till now that many of the bugs they are bad, and with also with the germ theory and things coming like that. But the concepts are changing rapidly since last decade maybe, and now we are thinking that maybe there are so many bugs which are friends, and so many bugs it might be neutral, and so many bugs which are actually essential to maintain our health. So here in this slide, we on one side we see so many diseases which might be uh, because of the disturbance of the microbiota, like the kidney diseases, various metabolic syndromes which we are facing badly today, diabetes, obesity, gastrointestinal disorder, to name a few. Then there is colon disorders and so on. And then there is various roles which are very important and which are played by the microbiota. They help in the immune function and maturation. They help in the development of brain and various behavior, and they also offer protection and also help in the clearance of various pathogenic organisms. So these are the new concepts which are developing recently. Now, there are some concepts which we need to keep in mind. Like if we talk of the microbiota, it is collection of all total organisms in a given environment, usually the gut or the skin or whatever. Then, so this is dealing with the live microorganisms. And then we talk of the microbiome. Usually it might be used interchangeably, but when we talk of the microbiome, all the genetic material which is contained together in the microbiota, that is called as the microbiome. Then there are few things which is eubiosis, which is a balance between the beneficial and the harmful bacteria. That is eubiosis. Then we have another term which is dysbiosis. It is an imbalance in the composition or the function of various microbial species which are found in any host. So if there is an imbalance, one species is becoming more dominant, so it will compete for the nutrition and it will utilize everything for its own benefit, and ultimately that will harm the host, causing lot of diseases. So there are some amazing facts and which we need to keep in mind, like 90% of various diseases differences in microbiome. Then if we talk of the microbiome, so their number is around 10 to 100 times that of the human cells. And if we talk of the genome, so our genome is only 1% and the genome of the bacteria is around 99%. So it might, and in some individuals it may be slightly lesser, but anyways, it may be at least 80 to 90%. So we have to see who is in control. I just talked that their genes are quite higher than our genes, and these microbial genes, they also turn on and off in are in response to various things, and they're turning on and off. They also turn our genes on and off, and in response to the various microbes which we have in our body. There may be changes in our body, and our body may become diseased, or even it may uh, progress to a very deadly disease like cancer if some particular genes or some particular microbial communities, not only single microbe, but various communities are pushing us in a direction which leads us to cancer. This happens in a way like there is a complex crosstalk. It is not the gut or the skin is not a place of complete silence. There are various chemical mediators which are communicating between the uh, microbes and the immune cells and microbes and the human cells. And this actually complex crosstalks between the bacteria and the bacteria and the host, they actually determine what is going to be the state of the host, whether he is going to be healthy or whether um, uh, he is progressing to any disease. So this is one very interesting figure where we see that if we feed not only us but also to the our bacteria, like if we have a lot of plant-based fiber-based diet, these bacteria, what they do, they are present in the lumen of our gut, and they produce lot of beneficial chemicals such as the short chain fatty acids, methane, acetate, and so on. So basically all these metabolites, what they do, they also manipulate the host gene expression, and they are not confined to the gut spaces. In fact, these metabolites, they cross the mucosal barrier and they reach to the bloodstream, and they can reach far away places, and having very prolonged and very um, great impact on various organ systems which are quite far away from the actual places where these bacteria reside. One more thing we should keep in mind, that our microbiome is dynamic. So there is some plasticity in this, and as we mature and as we age, usually this keeps on changing with the environment and with very other factors. It also changes with puberty. It also changes during the time of pregnancy. It changes as our diet changes. It might be a very standard American diet of type of thing, which is having lot of maybe high carbohydrate diet. It may be a vegetarian diet versus non-vegetarian diet. It may be a seafood based diet versus other diets. There are a lot of concepts which are emerging these days, like keto diet and Atkins diet and so on. Then there are medical conditions and various treatments which also change this microbiome. Exposure to pets is also a very important factor which actually affects the microbiome, and many more other factors are being investigated. So one thing why they are very important in the disease, because immune system also now has a this immunology also is gaining importance, that it is not only protecting against the invader, but in fact it is working together with the whole body system and different organs to store homeostasis. So in the maintenance of this homeostasis, these microbes are also very important. Like they train the immune system early in the life, and they help to distinguish self from non-self, and also to distinguish highly invasive organisms from the less invasive ones. Now again, the human microbiota modulates the immune system, and it also forms the surface flora, which may also, this surface flora also helps in defense against various pathogenic organisms. So they are also known as sometimes the guardians of the host. And we should remember that antibiotic administration and other drug administration also causes various dysbiosis, like intestinal, respiratory, and even in the skin. So what do you think, which microbiome should be the best? If we talk about, if we have a picture in mind, if we try to imagine a healthy microbiome versus a diseased microbiome. So what you see on the right side is a diseased microbiome. There is a predominance of certain species which are occupying and utilizing all the resources. Whereas if you see on the left, there is a very fine balance between all these various flora and fauna, and you have a nice, luxuriant, and thriving life of um, this ecosystem. So basically, our body is also like an ecosystem which needs to balance each and everything. Now, how dysbiosis may set in? There are many factors which can precipitate dysbiosis, like there might be a stress, there might be lack of sleep, genetic background, health status, various infections, inflammatory conditions, lifestyle habits. Environmental factors are also very important, such as diet, especially the high sugar diet and low fiber diet, administration of various drugs, xenobiotics, including antibiotics, food additives, and so on. Over hygiene practices also are said to play a role in the dysbiosis. Then there is eubiosis. So this, you can see a very important book which is gaining popularity in USA, and it says, this is one of the highest selling books. It says the beauty of dirty skin. So surprisingly, this is gaining very popularity nowadays. Like if your skin is dirty, it may not be disease skin. It might be a very healthy skin. Now we see that there are so many newer mechanisms of action of the drugs which are being discussed, along with the discovery of various newer bacteria in our microbiome. And it is amazing that there are 372 medications which might kill at least one of the bacteria species which are present in the gut. And out of these, 203 drugs were not antibiotics. So there are various drugs such as methotrexate, azathioprine, PPIs, calcium channel blockers, metformin, etc. And all of these drugs might not be harmful. In fact, some of the drugs, they mediate their beneficial effects with the help of modifying the microbiota. And these have been implicated in metformin, like how it controls the various metabolic effects, and also in case of methotrexate, how it is playing a role in the skin diseases by inhibiting various harmful organisms. This is another interesting picture. This is actually a fecal transplant, and it is not a hair transplant. So what happened actually, this guy who had alopecia areata, he underwent fecal transplant. That was of course not for alopecia, but that was for the C. difficile infection, which is the standard indication. And as a benefit, this guy also gained his hair follicles back. So this has been published in the case reports, and such type of case reports, they keep on coming. Now let us come to the skin disease perspective. Skin is very important since it is one of the largest organ of the body, and it has a very diverse microbiome. and it is not constant for all the areas of the skin. So various areas like the moist sites might have a different microbiome, which we can see at the right, like corynebacteria, proteobacteria, staphylococci, and so on. Dry sites also have a different kind of microbiome. Then sebaceous sites also, they have their own microbiome like corynebacterium, cutibacteriaceae, and so on. So uh, with this in mind, which microbiome is more important? The skin and microbiome both are crucial in maintaining the healthy skin. And one thing is very important, that diversity is the key to health. A diverse microbiome is always a healthy microbiome. Whether we prevent the dominance of any one bacteria, or maybe we can let all the various bacteria in particular area flourish. Nowadays, even in the skin diseases, the interest in microbiome is gathering pace. And as we see that skin diseases are usually complex in etiology, they are not that simple. If we leave 2-4 diseases aside, most of the skin diseases, they have a very complex etiology. And many conditions of them, they have a systemic component to them. And sometimes only the patient is focusing on the cutaneous manifestations, which are often due to the systemic inflammatory pathways. And such conditions, they also require a prolonged treatment, so they are very difficult for the patient and also for the doctor. And the another problem which confounds this is that patients are always looking for a quick fix solution. And as we all know that these therapies might also have some adverse effects, and because of this, sometimes the patient, they either they stop the therapy, or in the long run they may resort to other therapies such as alternate therapies. So deep uh, view in this is very enlightening if we talk of these diseases. Now there is another interesting thing. There is an emerging cosmetic this market, where they are showing prebiotic plus probiotic combinations, and they are also casting various skin products which are neutral to the microbiome. Okay, now I come to the various conditions where the role of microbiome has been discussed. I'll take them one by one. First one I am taking is the atopic dermatitis or eczema, and it is a long type of inflammation, long-term inflammation of the skin, and it is associated with increased risk of multiple comorbidities. So it doesn't come alone, and there is always some other allergy included with it, like food allergy, asthma, allergic rhinitis, and mental health disorders. Now, its etiology is multifactorial, and the pathophysiology is complex, involving a genetic predisposition, epidermal dysfunction, and T cell driven inflammation, which is possibly through the leaky gut as well. Now, there is one more thing, that there is a mutation in the filaggrin gene and the FLG gene, which is also causing impairment in the skin barrier, and which also contributes to its pathogenesis. It has been seen that increase in fecal Clostridia and higher Clostridium difficile and bacteroids ratio in this disease has been found. Another interesting thing is that there is a low diversity within few weeks of life of a child, and it confers a higher risk of developing atopic dermatitis later on in the life. Not only atopic dermatitis, but all various other kinds of allergies. If we talk of other species, Malassezia species colonization also increases with the severity of atopic dermatitis, and it has been detected in up to 90% of these lesions. Similarly, if we talk of the treatment, UV radiation exposure has been found to have a immunomodulatory effects, and somehow it also acts on the local flora and on the affected tissue, and it is B12 levels have also been linked to the severity of this disease, and it is potentially clinical significant improvement when B12 oral supplementation is given. And this also actually indicates that there is a gut dysbiosis which is going behind, and the gut bacteria, once they are normalized, gut bacteria, they start producing vitamin B12 and other metabolites, and which might help in restoration of the normalcy. So several probiotics, they have to seem to have a positive effect in treatment, and roughly they have a 20% reduction in the rate of this disease. And this concept is used in the biotherapy, and where the protective role of various commensal agents like Staphylococcus hominis, lugdunensis, and Staphylococcus epidermidis, they produce several molecules in that vicinity which are capable of synergizing the innate antimicrobial response, and they actually inhibit the growth of Staph aureus, and they help in improvement in the disease. Coming to another disease, I'll proceed all diseases in short, and finally then we will discuss the treatment. So coming to the cutaneous lupus erythematosus, it is a part of the systemic disease which is SLE, and it is often SLE is often considered a prototype of the autoimmune diseases. So coming to its etiology, it is heterogeneous and also a complex disease, and there is a adaptive immune network dysregulation which plays a very simple very important role, and there is abnormal T and B cell activation, there is self antigen exposure, and there is impaired apoptotic pathways, and all these contribute to autoantibody production in this systemic lupus. Then there is, if we talk of the disturbances in the microbiota, there is increased Glottia species and proteobacteria in the skin. And if we talk of the gut, there is in the Ellis types and the Odoribacter. Then some other studies have been shown that SLE related microbiota, they share some similarities across various geographic locations, and the patients have been studied from Spain and China, and both of them, they showed that there is enrichment of Probacterius species as compared to those which are found in the healthier counterparts. Then some other species like Rumenococcus nevus and Firmicutes, they also thrive in the gastrointestinal tract, and they promote the gut barrier impairment, and which is especially prominent in the SLE patients and those with renal involvement. So if these two bacteria are there, obviously this patient is likely to have a more systemic problems. Then there are not only anti-human antibody, but anti-Rumenococcus antibodies are also present in these patients, and the disease index score, like SLE-DAI score, it also has a uh, correlation with these two antibodies, RG antibody and anti-native DNA antibody levels, and it is negatively correlated with the C3 and C4 levels. Now, various antigens of another bacteria, such as R. intestinalis, they are also known to cross-react with the beta-2 glycoprotein 1 antibodies. So, one thing which is also playing a role is molecular mimicry of the human tissues played by these various bacteria which have been overgrowing. Now, when the SLE gut microbiota were replaced with the same proportions of bifidobacterium and a mixture of two Clostridia strains, various Rumenococcus and Blottia species, and they actually induced a Treg inducing effects, and they significantly reduced also the CD4 lymphocyte activation, and they changed the balance of the various cytokines which are responsible in disease, which are very, respectively the Th17 and the T helper 1 balance. Coming to another important disease, all these diseases are in fact very troublesome diseases. So now coming to psoriasis, it involves the formation of various erythromatous plaques and scaly plaques in various subjects. And again, it is formed due to the complex interaction between the genetic predisposition, the environmental factors, and immune dysregulation. And usually this triggers a rapid proliferation of the keratinocytes and followed by infiltration of immune cells. Am I there? Hello? Hello? Hello?
Yes, we can hear you. We can hear you.
Okay.
Good.
All right. So then there is a breakdown of immune tolerance to the cutaneous microbiomes, which has also been seen in the pathogenesis of psoriasis. There have been several studies which suggest the intestine is a possible origin in this disease and originating from dysbiosis. And it has been reported that the reduction of a few of the beneficial bacteria, such as Bacteroides and Akkermansia species, and there is an increase in the other bacteria, such as Firmicutes and the Actinobacteria. Now, various bacteria, if we talk of the skin flora, various bacteria found on the PV plaques, they also have Firmicutes and Actinobacteria and Proteobacteria. So, some other authors have also shown that there is a decrease in the good bacteria like the Actinobacteria and Bacteroides, and increase in Coprobacillus, Ruminococcus, and Streptococcus. Now, Staph aureus has also known to colonize the lesion in 60% of cases, and not only colonize the lesion, but they also start secreting the enterotoxins and even the TSS toxin. So basically, this imbalance is also visible here. Now, some other bacteria such as Firmicutes and Actinobacteria and Protobacteria, they also have been known. Then if we talk of the intestine, the altered intestine actually leads to bacterial translocation, like bacteria entering into the bloodstream and into the various epithelial areas and activating the immune responses, thus triggering inflammation and having a aberrant microbiome, thus uh, perpetuates these inflammatory responses. And there is another evidence that inflammatory bowel disease patients also have psoriasis in association, and the presence of Crohn's disease increases the risk of developing psoriasis up to 5 times. Now, patients that received mixed probiotics, that is Bifidobacterium longum and Lactobacillus, they had a decrease in the severity of psoriasis and a better response to treatment. And they also had a better response not only to the standardized treatment, but also a better response to vitamin D. This has to do something with the upregulation of the vitamin D receptors, which is which is not only playing a role not only here, but in various other diseases, that somehow these bacteria, they upregulate the vitamin D receptors. So thus these patients, they have a less need for steroids and they have a lower risk of relapse. Coming to another troublesome disease, which is acne, which is very common, and often the patient is under various stress, and it is also difficult for, usually because it is in the young girls and in the onset of maturity. So a lot of mental stress also is there with the disease, and this is again a chronic inflammatory disorder and of the pilosebaceous follicle. Then the acne has been largely associated with the acne's proliferation, and the load of this bacteria differ significantly between the acne patients and between the healthy patients. The various studies have shown this. Now, this bacteria actually triggers the release of pro-inflammatory cytokines after binding to the toll-like receptor 2, and they activate various other responses and T cell differentiation, and they actually recruit the lymphocytes and neutrophils to the acne lesion, thus playing a part in the etiology. Now, it has been seen that the loss of diversity between the various phylogenetic species of these bacteria, they actually the loss of diversity plays a role in the etiology rather than the proliferation of any all the bacteria when they are taken together. So if there are actually there are six phylogenetic types, and if any one or two are predominating and the rest are being left behind, only then the disbalance will trigger the acne. Otherwise, itself it is not that harmful. Now, if we talk of the Staphylococcus epidermidis, it actually inhibits the growth of this bacteria proliferation by favoring the fermentation of glycerol and releasing succinic acid. So it is also reducing the inflammation. Whereas when we inhibit S. epidermidis proliferation by keeping the environment acidic, so basically the acid-base balance also plays a role in the pathogenesis of the disease, And when in the follicle, when the sebum triglycerides are there and they are hydrolyzed, this is more prone, getting patient more prone for the disease. Now, various treatment options like intermittent pulse light, they improve the severity of acne, and actually this is thought to work by restoring the balance between the Staphylococcus epidermidis and the acne bacteria, and it inhibits the it also inhibits the sebum secretion. So various other therapies also, like the photodynamic therapy, they also increase the diversity of the microbiome, and they shift the follicular microbiome towards the epidermal microbiome and exerting its beneficial effects by inhibiting the pathogenic bacteria and modifying the composition and potential of uh, these microbiome in acne. Now, coming to the tools, how we, if you want to do a microbiome analysis, what all tools we can use. Like initially, it used to be very difficult, but with advent of molecular microbiology, this Herculean task has become simpler. Now, there are many methods such as 16S sequencing and whole genome sequencing. Now, they have demonstrated that various bacteria are present, like actinobacteria, they are dominating 36 to 51% are these. Then there are Firmicutes, which are around 24 to 34. Then there are proteobacteria and there are bacteroides. And these are the four major phyla which are found. Then it has also been suggested that many microbiota associated with humans have actually become extinct with the changing lifestyles of humans. And even some of the traveler or some of these microbiomes are yet to be identified. uh, Let us quickly see how we can do. Like there are various methods. First one is the culture based method. We all know that bacteria have been cultured since the times of Robert Koch, and it is actually a low throughput because one media will be required and then one sample can be done on one media, so many medias will be required, so much of time of incubation will be required. Then we talk of the other improvement, that is PCR panel. We can design various panels which have targeted various genomes in various genes in various organisms. So we can design a custom panel, and this panel also can identify only the targeted species. Unknown species will not be identified, and this, although is a cheaper, but it is a low throughput method, and the time taken is also quite less. This can be used like for the acne patients where you only want to look for 2 3 bacteria, and you can just when you suspect Clostridium difficile diarrhea, you can just target the bacteria with the PCR, and if it comes positive, you can go for the antibiotic therapy, or if patient is refractory, other therapies as well. Coming to the other method, which is amplicon sequencing, also where we do the commonly we do the 16S sequencing, where the partial genome, we are not looking for the whole genome, we are only going for the partial genome. That is usually the conserved regions are targeted. Conserved regions are those regions which are relatively constant in any particular species. So the usually the conserved regions are targeted, and then we target the other area which are the variable V3 V4 regions. There are various regions in this ribosomal area. Then we target the V3 V4 region. So the conserved region, we will identify whether bacteria is there or not, and with the variable region we will identify which bacteria is there with the help of the library, which you'll see later. So it identifies various organisms, and this is a high throughput. Large number of samples can be run. Like there are various sequencers, Illumina, PacBio, and so on. So we can run more than 300 samples in single run, and this run takes around 48 hours. And the cost is a factor in this thing. Like even though daily it is decreasing, but even today, if we talk of 300 sample, maybe 1.5 lakh, 2 lakhs, like that, there will be expense. Another thing is it just demonstrates the presence of the bacteria. We don't have the information about the functionality of the bacteria. So when we do the microbiome analysis, we should have the information of both. the information about the number of bacteria which are present, who all are present, and what are they doing. So we don't know what they are doing. Now, next method is the shotgun and whole genome sequencing. Here, not only we are identifying the whole part of the genome, but we are also knowing the sequence of each one of them. So, additionally, not only bacteria, but whatever is present, we can identify, like the parasite, the virome, the fungi, whatever is present there, and all the bacteria who all are present there, they can be identified, and whatever genes are there, they can also be identified. So, what happens like the function and the structure and the constitution, all these things can be studied at one go. But the problem is these costs are slightly more expensive than doing the 16s and other methods. Now you will be seeing, if you read some papers on microbiome, you will be seeing lot of pictures which are there, and sometimes difficult to guess what all is going on. So actually this generates a large amount of data, because any bacteria will have a large long sequence read, and usually it will, one bacteria will be in few genes, and there will be so many bacteria. So many number of GBs of data will be there. So for manually, it will not be possible to analyze this data. So there are specific bioinformatics tools, and you will be requiring a help of a bioinformatician to do the analysis of these things. And there are various softwares which are available, and the identification is only identification of not only the taxa but the genus also. It will be possible with the help of the database. So various database are there where we can compare this with the help of a software like Green Gene, Silva, etc. Then we have to estimate the diversity. There are various types of diversity like alpha and beta diversity. So how many bacteria are present in any particular host we are doing, and what is there in the particular population, and how these population they vary across the globe. So all different diversity analysis is also done. Then various tools for analysis are there like QAM2 and R, etc. Now if you see these two pictures, at the top we have a heat map. What this actually shows if you see here down below, all the this name of the bacteria have been written, and the darker shade actually shows that there are more number of bacteria. So according to the shade you will be able to tell. There is a reference scale there, and with the help of this you can see which one is increased and which one is decreased. Like on the bluer lines it will be decreased, and this is a log scale which has been shown, because of course the bacterial numbers will be in uh, millions altogether. So we are using a log scale. Down below, then you have a taxonomy heat tree, and here in the center you have all the bacteria, and the bigger offshoot, if you see, that means those bacteria are more in number, and then you have a diversification of that particular phyla, and individual bacteria will be, whatever is found out will be there in the as a branches. So it is just like a branches of a tree we are seeing this. Now coming to the treatment options. So this is very interesting. Like this branch is not only up to the research level, now it has come to practice level. And yesterday I was reading there are more than 200 various drugs or potential biologicals which are under development. And let's see how all these drugs they target. So they have a various category: probiotics, prebiotics, postbiotics. So there are various these things, and how they target, like they do health optimization by promoting the symbiosis, that is without harming the host, sometimes even protecting the host. So there is a whole chart which ranges up to from dysbiosis to eubiosis, symbiosis. Then there are various treatment strategies we see on the left side. Like there is a fecal transplant, which is one of the most drastic methods which can be adopted. Like it has been adopted in inflammatory bowel disease, in cases of colon cancer, in obesity, in sepsis, in C. diff, which is a standard practice there. And then where various probiotics they are being tried in various cancers, and along with the standard therapy they have been known Sometimes there are non-responders to various chemotherapies in case of cancer patients. So they convert the non-responders into responders. Then, of course, asthma, atopy, cystic fibrosis, and so on. Various therapeutic measures are there. So all these are known as the biotherapy, and it uses the protective effect of the commensal agents against the pathogens to improve the dysbiosis conditions in these patients. Uh, Here we are talking of the atopic dermatitis. Then this approach is quite different to what approach we have been taking. That is the approach of chemotherapy. Where the chemical agents are being used, here the biological agents or the bacteria and their products are being used. Various bacteria such as staph, various species of staph and their molecules, they are synergizing the innate antimicrobial response. So they are being used as a therapy. Now there are some probiotic creams which are there in the market and which help in improvement of the skin health. Then there have been approaches where the maintenance of skin microbiota homeostasis is achieved through the utilization of probiotics, prebiotics, and symbiotics. So let me elaborate these. Like probiotics are the bacteria themselves, the good bacteria themselves. They are the probiotics. Then prebiotics is the food for bacteria. So what happens, like usually the inulin and other high fiber compounds, they are added. It is the food for the bacteria. Then there are symbiotics, like two bacteria, three bacteria, they are acting together. So they are symbiotics. Then there are postbiotics, the bacterial compounds like short chain fatty acids, methane, and all, which actually modulate the immune response and the organ response. Then they will be the postbiotics. Now, microbiota, it also interacts with the host across great distances. So this is already known. So by giving these metabolites, like short chain fatty acids, various mental disorders and various other disorders can be treated. And now the various mechanisms are also evolving day by day, like where they are seeing that these actually metabolites are, they are acting as a ligands and they affect the even the cell immune cell phenotype which is developing, or which way the immune cell is producing. But however the exact mechanisms of these are yet to be explored, and they are as of now they are unclear. Now coming to the future implications. Now, as we know, as we gain better knowledge of the microbiota, there are several unanswered questions, and in the future, the research will guide whatever will be the next approaches in this. And one thing which we need is that understanding of the intricate interactions between the host and the microorganisms. And once new insight is gained, then there will be newer possibilities of developing various treatment modalities, and possibility of exploring these options in the treatment. Then there are antimicrobial treatments directed against various pathogens which are associated with skin diseases. They also eradicate the bacterial flora, the beneficial flora. And there have been efforts which have shifted to the maintenance of these microbiota homeostasis through various methods. The new thing which is coming up is skin microbiome transplant, that giving one person's healthy person's skin microbiome to other diseased persons, with a hope that this might cure the skin diseases. Then there is fast therapy. Then there are vaccinations are also coming up, like cutaneous vaccinations, which are against the these pathogenic organisms or the pathobionts, which are causing skin diseases. Now, there are certain limitations, like the cause and effect relationship is not properly known and it is still underway. The research is still underway. A lot of things are coming up. These organisms usually, they individually they have been studied, but there is more possibility of they having a community effect, because in many studies it is found that when these bacteria they work in a community, they have a crosstalk and then only they have a positive effect. Individual bacteria may not necessarily help in treating the disease. then the selecting the core microbiome is also challenging, because there are when you select any core microbiome which you want to target, there are several factors and several unknown factors which we might not be knowing, so it is difficult to target this. Then there are some unknown group of bacteria, and up to around 10% of the sequences which are being obtained, they do not uh, fall into any that of the known bacteria. So it is difficult as of now. We are yet to discover many newer bacteria and their roles. Now, till now, maximum of my talk was focused around that of the bacteria, because their roles have been uncovered quite more than those of the fungi and other microbiome. So the microbiome not only includes the bacteria, but also the fungi and viruses, and even the parasites. So the role of fungi and various viruses in this disease, that we don't know, and even the bacteriophages. So as we progress, there will be more knowledge about these. Concluding, now understanding the structure and function is an evolving field, and it is area with increased interest, and there is an important role in development of function and function of the human body, and it is also implicated in the pathogenesis of several diseases, and including that of the various cutaneous and skin diseases. Bacterial community manipulation is also an emerging treatment modality nowadays, and knowledge is evolving day by day with newer insights into the functional aspects. With that, I thank you all, and I'm open to questions.
Thank you, Dr. Sumit Gar. That was a very excellent lecture. And
Thank you so much.
like any new things, it takes time to assimilate and digest these things. So my feeling is that these things has to be pushed more often into the minds of the treating doctors or the persons concerned with this, because it is still very difficult for people who have been learning in and out from a long time that all the microorganisms are pathogenic and not helpful.
Yeah,
So this concept, once it gets into the mind, and we can think them as a friendly unicellular organisms for us too. So
yeah,
I
yeah.
think it just needs a push, and there are companies which are coming up with quite a new drugs, but my only concern with them is that, like, what they provide for gut microbiota, the cost factor is quite high.
Yeah.
So one who uses for a long time may not be afford to use that. Like it's almost 50-60 rupees a sachet, and 2 sachets per day if you want to give for the gut. For how long can you give? The person may not be able to afford that. So there's only concern regarding these things. So probably, if that is looked after, maybe the utility or use will be more often.
Yes, of course. And I was going through some of the protocols where people have tried manipulating the gut microbiome and the skin microbiome. The one thing which was there, like repeatedly and for a longer period of time, you have to give. Like if you give once daily, it may not work. For certain conditions, they are giving 3 times a day. Then only it
Yeah.
is working, and sometimes they are also giving it along with the antibiotics. Like if you want to eradicate the C. difficile, so the standard care is already being given for C. difficile, and then along with that they are giving probiotics to eradicate that particular bacteria. It is like that.
We have been
And
trying on
sometimes,
and off. Yeah, please go ahead.
sir, and sometimes they are also using bacteriophages along with that. So bacteriophage will target the harmful ones, and now the probiotics and prebiotics, they will try to reestablish the microbial balance over there.
We are not trying this in cases of atopic dermatitis and chronic urticaria, uh, but our confidence level is not developed, to be frank, so much.
Yeah, yeah, yeah.
Dr. Rawat, very nice lecture. Actually, my question is that, uh, as you told that the
Yeah,
gut microbiome is very dynamic and very individual to individual.
yeah,
So,
yeah,
how you can provide a specific therapy if someone is coming with acne, or like the two person, how you will prefer which type of therapy you will give? How you personalize the gut microbiome therapy for the future?
Okay, yeah, nice, very nice question. Actually, uh, these things are really under development, and what is only few protocols have been developed. Like if you talk of the acne, like our basic thing is to restore the balance. Like what I showed, like um, in acne, we are not giving this microbiome therapy. What we are targeting is to restore the balance. So through laser therapy and through UV irradiation, what we are doing is we are trying to eliminate the pathogens, and we trying to balance the this thing, the skin microbiome. And if you talk of the other situations, uh, there are certain bacteria such as lactobacilli and this there was a very nice experiment where they were giving species of Saccharomyces and Lactobacilli. So what happens that what I just told, that bacteria, they act in a community. So this thing was very interesting. Like they gave only two bacteria, and later on what they did, they again I don't remember the exact study, but anyways, what happened, like after this they again did the sequencing. So what happened? They had only given a mixture of two bacteria, and these two bacteria, they actually created a positive environment for other bacteria, like the Faecalibacterium prosnitzi, which was there, and there were Akkermansia species, which are known to be beneficial for health. So what happened? Lactobacillus species, they actually helped the Akkermansia also to grow. So the diversity initially was maybe just the 10 species. They gave two species, and later on they saw 30 species. So these two species are acting in a crosstalk with other bacteria, and they are helping each other to maintain a balance. Like if you are giving, it is you can uh, maybe imagine this in a way, how you manage your garden. Like if you have uprooted all the harmful cactus and all, and if you have put some fertilizer, and if you have put the if you have made the soil, and if you have put some seeds, some other seeds will always be there, and with the favorable environment, those plants will also grow, maintaining a balance in your garden. So it is the gut situation is also a similar situation.
Dr. Rawat, thank
Sir,
you very much. Very detailed, presentation. And now, in terms of, already it has come into clinical practice. Okay.
already, yes.
Whatever, we were not waiting for complete understanding of mechanistics. Okay.
Yeah, yeah,
Mechanisms.
yeah.
But
Yeah.
it has come into clinical practice. And what is the side effects, do
Yeah.
you have any account for that, any publications? What type of
Yeah.
side effects is expected,
Yeah, actually, sir, this fecal microbiota transplant has been used with caution, because there have been several incidences, like there have been incidences of transmission of hepatitis, maybe hepatitis B and C in few patients, and there are some other things. And there was one incidence where the mother was a thin mother, and her daughter was very fat, and mother
Oh,
was suffering from C. diff diarrhea. And what happened, the daughter was obese, and she received the microbiota from the obese girl from her daughter, and ultimately the
oh,
mother also gained a lot of weight. So things have been like that, and even incidents of death have been also been reported. So especially in cases of ulcerative, some cases of ulcerative colitis, not all. So it is used with caution in cases of ulcerative colitis, whereas in C. diff, it has been very useful. In Crohn's disease, it has been useful, but with in ulcerative colitis, they actually they have caution. Now there is one more interesting thing, that there are some super donors. Like some people are very healthy, or they are said to be super healthy. And
oh,
China is, I was studying, China has encouraged people to donate their stools, and they freeze their stools and keep there, and they are paying people, they are paying money to get the stool refrigerated. And these super donors are very happy, because their stools, they contain large diversity. The more,
oh,
so now there is a concept of internal richness versus external richness. Like internal
oh,
richness is, I have like maybe 200 bacteria, so I'm very
oh,
rich, so I can donate my stool and earn money. and some other guy may be a very rich guy otherwise, but he doesn't have any bacteria, so internally he is very poor.
So the number
Dr.
of
Rawat,
bacteria.
sorry.
Yeah,
Dr. Rawat, one small query in this. When you
yeah,
talked about the super donor, I had read
yeah,
something not in terms of the gut microbiota or the gut patients, but in
yeah,
general, when it comes to super donors,
yeah.
what are the criteria that they choose? Is it a specific to the disease, or is it specific to the people? What is it chosen? How do they identify them?
Okay, actually, it's a slightly difficult question for me. But as far as because I am not dealing with the this thing stool bank and all, there are stool banks. I think there are two stool banks in India, but there is a general screening procedure. Like you have to screen for the age, for the disease. Patient should not be overweight. You should not have diabetes. You should not have metabolic syndrome. No liver disease and all. You should be HIV, hepatitis B, hepatitis C, all screening should be done. So all these criteria are like that. You can't take stool from very old individuals. So the weight criteria, the height criteria, the age, and all those are there. ANA negative, and all those testings are being done.
So it's
So
a quality check which is being done before they collect, right?
yeah, there is a questionnaire, like there is a physical quality, like the questionnaire, whatever, based on the questionnaire you can do, and then later on there is a testing as well. And when they donate one particular, donate the stool from one particular patient, then they will be sorry, not the patient, the donor. When they get this thing, they will be able to evidence the benefits in the other person. So what I read in this one was some donors, they actually have a very good effect on the recipient, whereas visually or on table or on paper you might appear very good, but in practice that stool does not work. So these super donors have been chosen on those where the stool is working better.
Okay, sir. Thank you so much.
Yeah.
Good.
Okay, most welcome.
The
Are there
cost,
anyone?
cost you are talking,
Yeah, yeah.
targeted sequencing,
Yes,
and
yes.
300 samples, you said 1.5 lakhs.
Yeah, sir. Patients, if sorry, some people they actually quoted in one lakh. Uh, I think $150,000 they quoted like that in US. One talk I saw.
Per person.
No, no, not per person. They at one go they sequence 300 samples in single
Okay,
this thing.
300 samples they quote for that. Okay.
Yeah,
That
yeah,
comes
yeah.
to 50 rupees per sample. 300
300.
divided
Yeah,
by
yeah,
1
yeah, yeah.
lakh.
Yeah, there is. I'm sorry, there is some calculation mistake.
Here, what happened here.
Actually, here
Yeah.
I did some
Okay.
samples. I told you, like it was
Uh,
around 8,000, 9,000 for one sample. If
yes,
we,
yes, yes.
yeah,
Targeted is like that only. 7,500, 8,000, like the range, You are
yeah,
right. And see, in terms of dysbiosis, my
yes, sir.
question is dysbiosis. We are not going to specifics. Okay.
Yeah, yeah,
What
yeah.
is the percentage? Okay. From that point of view, targeted is enough.
Yeah, for dysbiosis, we can demonstrate with the targeting because we are not
Oh,
looking for any functional aspects of the
yeah,
study.
yeah,
Yeah, yeah.
yeah, yeah.
And
Now,
main
therapeutic.
thing is diversity. So
Yeah,
diversity we will be able to tell with the help of this. Even though some
yeah.
species we will not be able to find up to some bacteria, we will not be able to demonstrate up to the species level. But even up
Okay.
to the genus level, if we identify, it will be sufficient.
Good. And based on this, in terms of therapeutic intervention, is it supplement, is general supplementation or specific, which is lacking in an individual? Bacterial supplements, what we have
Yes,
on
sir.
the market, there are
Yeah,
different
yeah,
types of compositions.
yeah,
That is, generally they produce that. But if I have a patient, I know what is dysbiosis, I know
yeah,
which is lacking. Can I
yeah.
give that lacking, whatever is lacking, those bacteria? Is it possible?
Yes, sir. It is possible, but as of now, things are under development, and there are two things. One, actually, why this therapy directly came into existence, I was studying this. Like there have been studies of safety. So safety
Oh,
is one thing. There have been study of safety. So these formulations, they are safe. So as far as they
okay.
are safe, they can be given. Another thing is efficacy. So efficacy of none of these have been proven. There are various studies on efficacy. So how much efficacious and in whom it is efficacious, this has to be documented, and it will take a long time. So as of
Yeah,
now, as long as it is safe, it can be
yeah,
tried. And if
right.
patient is getting some benefit, so we can see that at least there is some improvement along with the standard of care. So one
Okay.
rider was there, like you can't deny the standard care as of now, that you
Yes,
stop the standard care treatment and then you start the other treatment. That is not advised. It
yeah,
is only like with the standard care, if you give this also, it has a add-on effect, may not be in all the patients, but at least in
right.
30-40% patients you will have a long lasting effects, you will have a better effect, you will have a better control over the disease. So this is what
Yes.
the influence is.
Microbiota functional nutritionists. Nowadays, we are listening this
Yes,
functional
yes, sir.
nutritionists, and
uh,
what they do, they only give nutraceuticals.
Nutritive. Yeah,
Huh? Yeah.
yeah,
Okay.
yeah.
Good.
So, basically, functional nutritionist and functional medicine is like basically focusing more on the physiology.
Yes.
Initially, the approach has changed. Basically, initially, just like I talked, like initially approach was more on the chemotherapeutics, like the chemical agents. All the
Yes.
medical stores used to house lot of chemicals or lot of extracts, but
Yes.
now we are focusing more on the biological factors like the
Yes.
enzymes and the bacteria and so on. So
Right.
basically, by having a deeper understanding into the physiology and the function, so we are trying to improve the function of the body with the help of
Right.
these agents. So
Right.
that is targeting these things. And lot
Right.
of other with nutrition, you just touched on upon it. So with nutrition, what we mean is like nutrition not only for the host, but nutrition
Okay.
also for the microbiome.
Right.
That is very important. So when you give a nutrition for the microbiome, then only you will have a healthy microbiome.
Healthy microbiome.
And once you go towards a dysbiosis, your dysbiotic bacteria will produce signals which will produce some cravings. Like if you talk of high sugar diet, and if you are taking sweeteners, study has been done with artificial sweeteners. So when you
Okay.
are taking artificial sweeteners, what happens? Certain bacteria, they give you a craving of these, and you are more likely to drink the tea. If you are taking cups of tea, then tomorrow you will take 3 cups. Maybe after that you will take 5 cups of tea with the sweetener. So they will
Okay.
predispose you to have it more and more because they
More
like
only.
the sweet taste. Yeah. So and you will have more dysbiosis that way. So you should have the food for maintaining the diversity. Foods
Yeah.
like nutraceuticals, like even the foods which have phytochemicals, various like curcumin and then coffee and so many other phytochemicals have been discovered. Like even in the green tea, there are phytochemicals which actually enhance or which actually help these bacteria to grow. So that
Okay.
is why. So initially there was only a concept in nutrition. There was macronutrient and macronutrient. There were no phytochemicals. There were no other categories. But now there are phytochemicals as well. So these chemicals, they actually help the bacteria also.
Right. Thank you.
Yes, sir. Okay, most welcome. Any questions from you,
Any
sir?
other questions, please? I think it was an excellent one. We all are stunned by all the new entities. So probably
My
yes,
pleasure.
we will listen more and more, and we'll get used to the concept of being internally fit.
Yes, sir, of course.
Thank you, Dr.
Internal
Rao. It
richness.
was an
Yeah.
it was an
Yeah.
excellent lecture. Thank you.
Yeah.
Thank you, sir.
Gargi,
Sir, I
yes.
have something. I have something to say.
Yeah.
Can I ask one query, sir?
Yes,
Yeah.
please.
Sir, there are trillions of gut microbiome in our body,
Yeah.
and there are around 1400 species which are harmful to human body as such. So now we are talking about the dysbiosis. So have you come across how many bacteria are there which are harmful for the skin diseases? Like we have seen, there are, for COVID-19, there were two bacteria found out by the scientists, that is Cutibacteria and Lentimonus. These two bacteria, they are harmful. They were harmful for the severity of the COVID-19. So, have you come across for which, how many uh, microbiomes are responsible for the skin diseases?
Look, this is, I think, a very complicated question, like how many bacteria have been there. So the research is still going on. Like just you told, I haven't heard of this research, but there are, of course, researches where newer and newer bacteria are being discovered day by day. But
Yeah, exactly.
the point is not to discover roles of these bacteria here, but the point is to maintain a balance. Even if they are present, their mere in one of the slides I showed, that mere presence does not indicate that they are causing a disease. They may be present there and without causing the disease. Once they get a dominant role and once there is a opportunity, then only they will be able to cause the disease. Like COVID-19 was one of the opportunities for them. So
Yeah,
basically, they are playing as an opportunist. They are not necessarily hardcore or they are not a sure shot pathogen, just like the Mycobacteria leprae or maybe the Mycobacterium ulcerans and others like the Staphylococci. So these are actually known pathogenic, but there will be so many pathogens. There will be, like, if I was one day I was talking to one of my friends who is dealing in ICU practice. So nowadays in ICU with so many immunosuppressive therapy to kidney patients and all, every time you get a new bug and you are bugged by the bug itself, and you don't know its identification, then you have to go for sequencing and then you identify, oh, this bug has been isolated. So every day there is a new environmental bug. It gets an opportunity, enters inside, and it will produce a disease. But the problem is we should not give an opportunity by maintaining the balance. That is the crux of this.
The best news what I got from this whole concept is that the number of microbiota, that it is about more than 3.5 times your cellular level. So that means
Yeah.
I actually weigh 3.5 times less than what my weighing scale shows. So I'm happy with that.
Amazing.
Thank you, Dr.
Yes.
Gar. It was a nice one.
Thank you, Dr. Rawat. Thank you so much.
Okay, nice interacting with you all. Maybe we can participate in more discussions like that. And hope
Sure,
my
This
presentation was
interesting.
not too long.
this
No, it was
person,
interesting.
Dr. Urade, who spoke just now,
yes,
Yeah.
sir.
only on yesterday, day after yesterday, we were in a meeting.
Yeah,
The
Okay.
ministry called a meeting. This man is going to map microbiota among primitive tribes.
yeah,
That's great. That's great.
Onges, Jarvas, and
Yeah,
in
yeah, yeah.
bones, bills.
That's
This
great. Actually, Indian
yes,
microbiome has
we are on
not
the
been
way, sir.
cultivated.
We
Okay.
have not yet
uh,
started, but it is we are on the way
it
to
is
do
going,
it.
Mhm.
they are going to
Now
start.
there was one more team from DBT which was working on this thing in
IGIP,
various population. Yeah,
IGIP,
yeah,
IGIP is working on that.
yeah,
Yeah,
yeah, yeah,
DBT. Yeah, right.
yeah, yeah.
He's an anthropologist. He
Ah,
wants
that's great.
population mapping, something, these people do.
Yeah, yeah, yeah.
That sampling matters, sir. Proper sampling matters
Yeah, definitely,
in such
definitely.
type of
Actually,
studies. Yeah,
yeah, some DNA that I will suggest, some DNA stabilizers are there which could
that
be
we are
used.
doing.
Like,
That we are doing.
yeah,
That
yeah,
we are
yeah,
on the way to do it.
definitely.
Considering only the PVTGs first of all,
Yeah,
and the pilot survey we are going to start this month.
that's great.
We will start
All the
from,
best.
sir. We will start from Savra community.
Wow.
Yeah,
Right. This is anthropological survey of India. They survey all humans, you know.
Yeah.
Right. Gargi, what are
Yes, sir. Yes,
thanks?
sir.
Yeah,
Yes,
please.
sir. Yes, sir. A very, very interesting session, sir, because I myself have so many questions more coming up, but no, not right now. This is, I mean, it's too big a platform to ask such type of questions, and then they are also having many more leading questions. Some other day,
Yeah,
I will rack your brain, sir, to ask more questions about this. But this
of
is a
course.
very good starting point because every time you hear something new, we're hearing about microbiome, we're hearing about how it is going to be useful. But the fact that it can be used and how it is going to be used, when you explain that, that was what was very interesting, that we are able to understand what it is. It's not something like, it's something totally unknown. It is a known
Yeah,
concept. It just needs more exposure, it needs more understanding, and for more people to accept it. And obviously there are some drawbacks, there are some advantages, but obviously in any technique that is there, so we have to consider all of that and then go ahead for it. But a really very good interactive session with a good discussion from everyone. I'm glad we could do this. Maybe we can have something like this again for the future also. Thank you, everyone. Thank you, Dr. Lahiri and Dr. Rawat,
Thank you.
for taking the session,
thank
and
you.
thank Professor Rao for convening the entire thing. And thank you for all the other participants also, because without you, this event wouldn't have been able to take place, sir.
Definitely. Thanks to all the viewers.
Thank you, Dr.
Thank you.
Gargi.
Thank you. Good
Thank you
night.
so
Thank
much.
Thank you,
you.
sir. Good
Bye-bye.
night.
Good
Bye
night.
Thank
bye.
Good
you.
night,
Good night,
everyone.
everyone.
Take care. Have a good
Take
day.
care.
Yeah.
Good day.
Okay, sir.
Okay. Good day.